Hello everybody, welcome to our video lecture on basic skeletal muscle structure. Kind of a review of some material that you really should know from anatomy, but it's absolutely essential to your understanding of the physiology. So here we have a picture of a skeletal muscle. Notice that this skeletal muscle is attached on either end by tendons to bones. Okay, now if we take this skeletal muscle and we open it up, we look inside of it, basically what we're going to find in here is that the muscle is going to be composed of a number of bundles. Those bundles are going to be surrounded by layers of connective tissue. Now these larger initial bundles here that you see here and here and here, these are called fascicles. Fascicles in muscles can be arranged in different ways to produce muscles with different strengths and different weaknesses. Now the entire muscle itself will be surrounded by a connective tissue layer around the outside called the epimysium. Each one of these fascicles inside the muscle is going to be surrounded by its own layer of connective tissue called the perimysium. Now if you take a look closely at one of these fascicles here, like your textbook pulls it out here, you'll see that that fascicle itself is a bundle. And basically inside that fascicle you're going to have a bunch of these individual muscle fibers. So a fascicle can be defined as a bundle of muscle fibers, and a muscle can be defined as a bundle of fascicles. Now let's take a closer look at a single muscle fiber or a single muscle cell. Before we change the slide, notice from the image here you can see that this muscle fiber is basically going to be a single muscle cell that can be very long, that can basically extend the entire length of the muscle from tendon to tendon. We're looking at just a small piece of a muscle fiber in this slide here. So when you're looking at your muscle fiber or your muscle cell, the first thing to look at is the plasma membrane of the muscle cell, which you can sort of demonstrated for you here in gray. Now technically the plasma membrane of a muscle fiber is called the sarcolemma. A sarcolemma is the plasma membrane of a muscle fiber. Okay. Now that plasma membrane actually has these folds in it. And these folds actually form these tiny tubes that sort of pass through the center of the muscle cell and create this sort of network of tiny tubes. Those folds of the plasma membrane are called transverse tubules. It's very important to notice that the transverse tubule is technically the same or is connected to the plasma membrane or the sarcolemma on the outside of the muscle cell. Inside muscle cells, you're going to see lots and lots of mitochondria. These muscle fibers require lots of energy to perform their functions. Okay. Also in muscle fibers, the cytoplasm has a special name. The cytoplasm of muscle cells is called the sarcoplasm. This is kind of going to be a theme that you see here with organelles inside the muscle fiber. They're going to start with this term sarco. Sarco means flesh, and so whenever you have your pound of flesh, you're holding a pound of muscle there. Next we have these blue organelles, this blue sort of network of membranes. This is the sarcoplasmic reticulum, or the SR. I am kind of notorious for just abbreviating sarcoplasmic reticulum as SR, and so you should be familiar with that. Now, one of the things that's going to be very important about the sarcoplasmic reticulum is that the sarcoplasmic reticulum is going to have primary active transporters called calcium ATPase pumps. those calcium ATPase pumps are constantly pumping calcium into this sarcoplasmic reticulum. And so this sarcoplasmic reticulum stores a large amount of calcium for the muscle fiber. Last structure that you're going to see here in the muscle fiber are these structures that kind of look like cylinders all over the place in here. These cylinders are actually made up of protein filaments, basically long interconnected protein fibers. These cylinders of these protein fibers are called myofibrils. And myofibrils are going to be really important because these are going to be the structures that will be shortening during a contraction of this muscle fiber, this muscle cell. It's also important that you note, though you don't really see it in this picture, that muscle fibers are very long, very large cells. And one of the things they get from their development are they're multinucleate. They have many nuclei throughout the cell. Now, what we want to take a closer look at are regions that you see here, where you have a region of sarcoplasmic reticulum, then a region of T-tubule, then another region of sarcoplasmic reticulum. Those specific regions that you sort of see highlighted out for you here are called triads because it has the one region of SR, the T-tubule in the middle, and then another region of SR. Now, like you see in the image here, these regions are actually really close together, so close in fact that there are proteins in the membrane of the T-tubule that can literally reach out and attach to proteins in the membrane of the SR. Now, the last thing to notice about the muscle fiber here is when you take a look at the organization of the T-tubules and of the sarcoplasmic reticulum and the triads, you'll notice that they're all arranged around these myofibrils. And when we get to the function of these muscle fibers, that will become important. So what we want to do to understand how muscle fibers contract is we want to take a closer look inside the myofibrils. A good way to describe a myofibril is to describe it as basically a number of these structures that you're seeing on the image here called sarcomeres, these protein filaments. And in the myofibril, these sarcomeres are arranged single file or end to end. Okay, so if I pull out one of these sarcomeres here, okay, I'm going to see what I see on the screen here. So here we have an individual sarcomere. In the center of the sarcomere, we have a anchoring protein called anchoring protein filament called M-line, and at the ends of the sarcomere, we have kind of this zigzagging anchoring protein called Z-line. We have a Z-line on the other side of the sarcomere here as well. So technically, these Z-lines are going to be the boundaries of an individual sarcomere. One sarcomere is everything between these two Z-lines. Now, attached to the Z-line, we have these purple chains that you see here. This is called the thin filament or the actin filament. Branching off the M-line here in the middle, you have this thicker protein filament that's in pink. This is called the myosin filament or the thick filament. Attaching the thick filament here to the Z-line, you have this protein fiber that looks kind of like a slinky. This is called titin. Now, during contraction, this sarcomere is going to sort of shorten. During relaxation, this sarcomere lengthens. So you got a lot of movement of this sarcomere. And what that titin here is going to do is it's always going to reset this sarcomere to an optimal length. Now what we want to do next is zoom in on these two filaments, the thick filament and the thin filament, and we want to take a look at these protein filaments in a little bit more detail. So we can see the detailed images of these particular filaments in the image that we have here. So let's start with a thin filament here. Notice if you take a closer look at the thin filament, what you're going to see is that the thin filament is composed of these two chains, each of them kind of looking like a string of pearls, where each one of these little spheres here is a pearl and these two little strings of pearls. Each one of these little spherical proteins is called a G-actin. The G in G-actin stands for globular because of its spherical shape. When you link all of these G-actins together and you form these two chains, we call this F-actin. The F in F-actin stands for filamentous actin. Now if you notice here underneath the laser pointer, on each one of the G-actins there's going to be sort of a dark circle there. Each one of those dark circles is a myosin binding site, and every single one of these G-actins has one. The last proteins that you're going to see here on the thin filament are going to be these regulatory proteins that are going to regulate when contraction occurs and when it doesn't. So the first one is this blue one here called tropomyosin. The other one is the pink one here called troponin. Now troponin is a calcium binding protein, and whether or not calcium is bound to troponin will affect the position of tropomyosin. So troponin essentially controls the position of tropomyosin. Now, when troponin is empty, like it is in the image here, tropomyosin is actually going to sit right on top of the myosin binding sites, and it's going to block them and prevent myosin from binding or interacting with actin, like it would right here if tropomyosin were not in the way. However, when calcium binds to troponin, this will cause tropomyosin to move off those binding sites, move out of the way, and then it will allow myosin and actin to bind to each other. When myosin binds to actin, this event is very important. It's what's called a cross bridge, and those cross bridges are going to be extremely important. Let's move on now to the myosin protein over here. So the myosin filament is here, and if I were to pull out a single one of these myosin proteins, the myosin protein looks a lot like a lamppost with sort of a heart at the tip of it. So you can see that general shape of the myosin protein down here. Now what I'm going to do is I'm going to take my pen and I'm going to break my myosin protein into three pieces. So the piece here that has the T is called the myosin tail. This region of the protein attaches to the rest of the myosin filament. It's basically structural in nature, and we're not really going to talk about it very much anymore. The next region here is a flexible region called the myosin hinge, and it can change its shape. The final region is this heart-shaped region here. This region is the myosin head. And I apologize for my inability to draw too well with this mouse, but this region here that looks like a heart is called the myosin head. Now the myosin head, like the picture shows you, is going to have two binding sites on it. It's going to have a binding site for actin at the tips here, and it's going to have ATP binding sites kind of off to the side. Now, it's these ATP binding sites in particular that are going to determine the position of this myosin hinge. Remember, I told you the myosin hinge is a flexible region. And so what position it's in is really going to depend on what is bound into that ATP binding site. So to show you how the ATP binding site affects the shape, I'm going to show you the two basic shapes of myosin up here at the top of the slide. Now I'm going to draw the myosin hinge with sort of dashed lines like this. So you can have the myosin hinge in a position that I like to call the relaxed position. This is a low energy position, and that myosin hinge is kind of up at an angle. It's kind of bent. Okay? And I'm going to draw the ATP binding site right there. The other shape for the myosin protein is this one here, where that myosin hinge is actually laying down flat. This is what's called the energized state, and so we're going to draw an E next to it. That's the energized state. I'm going to draw the ATP binding site right there. Now, like I said, the position of this dashed region, which is our myosin hinge here, depends on what's bound to the ATP binding site. If the ATP binding site here is empty, or if it's bound to ATP, the myosin head or myosin hinge is going to be in the relaxed, low energy position. However, for this myosin head, or myosin hinge, to be converted to this energized state, to this high energy state, you have to put energy into it. And one way that you can put energy into this protein is by taking this ATP and hydrolyzing it, breaking it down into ADP and inorganic phosphate. Remember, when you do that, you're going to release energy, and that energy is used to set the myosin head in this high energy position. This is kind of like setting a spring. When you have a spring and you squeeze the spring, that spring has energy, so that whenever you let go, it automatically snaps. Another analogy to the myosin head is to think of a catapult. So if you have a catapult, right, the catapult has two positions. It has a high energy position like this, and the catapult also has a low energy position like this. So the high energy position, if you have some unlucky guy here, sitting on your catapult in the high energy position, and then you throw that switch and you let the catapult sort of use up its energy, right, or release its energy, it's going to fling to this sort of relaxed position, and it's going to send this guy here flying through the air. Okay, but once you've sent this guy flying through the air, if you want to go back to sort of throw maybe somebody else through the air, you're going to have to use some energy, right, to reset this catapult. The same thing is true with the myosin head. The relaxed state that you see here is what you're going to have after a contraction has occurred, and so it's going to require energy to set this myosin head back to this energized state so that it's ready to do a contraction again. When the myosin head binds to the actin filament, it's going to pull on this actin filament, and it's going to pull it closer to M line. Now, when it does that, this is going to shorten the sarcomere, like so. That shortening of the sarcomere by myosin pulling on actin is called the sliding filament theory. And this sliding filament theory is how people believe that a contraction occurs. So myosin pulls on actin. This shortens the sarcomeres. Those shortening sarcomeres shorten the myofibrils in the muscle fiber. with
the
shortening
a real picture
myofibrils in the
of the
muscle fiber.
sarcomere,
So you can see
the
two
muscle fiber
sarcomeres
itself.
here,
And
and then
then
you've got a
shortening
Z
muscle
line
fibers are
here
going to
and a Z
shorten the
line
entire
here and a Z
muscle.
line here.
They're going to
So that
pull on
means you're
that
going to have
tendon,
one sarcomer between those two
and
Z
this is going to
lines,
generate a
and one
force that we
sarcomer
measure as
between
a
these two
tension
Z
here.
lines. Okay, your M line is right there in the middle.
If you want to see another
So
view of
during a
this sliding
contraction, what's going
filament
to
theory,
happen is this myosin is going to tug on this actin and pull those actin filaments closer to M line. And you'll notice that this is effectively shortening this sarcomere. Now when you look at this process occurring in a myofibril, so what do we have here between the two brackets there is sort of an individual myofibril. And in the myofibril, you can see your individual sarcomeres. So right there where these two red lines are here and here, those are Z lines. And so everything between the Z line here is a sarcomere. So the M line is the purple line right there. The thick filament is in purple. The thin filament is the light red. So when each one of these sarcomeres shortens in that myofibril, you can see that the whole myofibril shortens. That will shorten the muscle fiber, and that will shorten the entire muscle. Okay, so hopefully you enjoyed this lecture and you'll review it before we talk about the details, molecular details of muscle contraction.